Firstly, if I could introduce Ian Carmichael, who is the Technical and Applications Leader for Cytiva Asia, an experienced biotechnologist with over 20 years in the industry and a long list of experience, as you can see. And he's been with Cytiva or GE since 2015 and is currently the Bioprocess Technical and Applications Leader in Asia. Then we have Vasu, who is a technical leader, also in the Asia office, a biochemical engineer with over 14 years experience in the industry. And he is the technical leader for Upstream-Asia and has been with Cytiva since 2016. Last but not least, we've got Jai, who is a digital service engineer, and he's has over 11 years experience in automation. So he leads the new digital initiatives for the APAC region, providing expert solutions, and has been with the company since 2014 and is, and no guesses as to what he'll be sharing with you. So without further ado, may I ask Vasu to share his slides, and then he can start. We would also welcome you to ask questions after each person has presented. You can either unmute yourself or enter them in the chat box as the presentations proceed. And also there'll be an opportunity at the end of the session to ask any of the speakers any burning questions you may have. So over to you, Vasu.
Thank you. Welcome all to the session, and I hope you and your dear ones are in good health. So without ado, I'll get started. So the first half, first 30% of the presentation would be more of upstream, and then followed by downstream, and then the automation. So that's going to be the format. So what we'll do here is in the upstream perspective, what I'm intending to do is to take it stepwise. So understand need, what are the key drivers, where is the trend, and we'll look at strategies for perfusion processes or continuous processes in the upstream. Then we'll deep dive a little further to see what's important at all the minor unit operations that are involved in the upstream processes, like be it the bioreactor, the chamber, the perfusion system, allied systems like mixers, etc. So what is that? What are the considerations that we need to do? And then I'll end up on the key factors that we should be considering in designing a perfusion process. Yeah. So for everything starts with the need, right? So what's the need of the, why do we need perfusion? That's where everything starts. So we know with increasing population, we have market also growing alongside, and that we need to answer certain questions. So the demand is increasing, so we are not able to cater, or the production capacity has its own glitches or crunches, I would say, and the need to have every medicine available to every corner of the globe. So that's where you need improved and high cell density processes coming in. So other reason why you need this is two things, I would say. One, the necessity to have improved processes. So you need improved processes. You no longer rely on traditional processes, and also on the novel diseases that we encounter. Like nobody knew that the entire world will be locked down for 2 long years due to COVID, right? So that's also bringing us into a mode where we have to come out with solutions faster. mRNA, for example, was existing, but we see that getting the limelight only during the COVID time. So those are some things that are there. And what's the uncertainty when we talk about this? We do not know, for example, for a specific product that we intend to do, what's the market demand, how would the clinical efficacy look like, what are the different types of approvals that we need. We know regional approvals are there. Europe has their own approval, China, US, every country have their own regulatory approval methods. Are we going to compete with giants which have multiple manufacturing facilities across, or it is just going to cater a specific region? So all these are some uncertainties, and sure, we have been always answering such uncertainties, without which we wouldn't have been here with all these many biopharmaceutical products. And also, not the least, but the cost pressure. I would not say it's just the cost. It's both the time and the cost pressure that we have. The faster to the market, speed to clinic, speed to market. It's something kind of saga that in the recent times we see. We know this, right? I've written biosimilars here. The first one to come out with a biosimilar molecule in the market after a patent expires, gets the maximum market share. So that's kind of a thumb rule now. So it's important that we come up with processes faster. So what are the key drivers, right? So if I have to, fit in all those options that we have into 4 big pillars, first is the facility, I would say. What's important with the facility? Facility is where you start, right? So that's the starting point. So you need to design a facility which is flexible enough. When I say flexible enough, think of what kind of a facility you need. Are you looking at multi-product facility? Are you looking at catering such huge demand product in the market, right? So these are some things that we need to look at. Apart from that, when I say regional manufacture, develop and build a facility or a process that is so flexible that you can adapt it to any region, right? So you're developing a process in a stainless steel bioreactor. It should be so adaptable that you give the process to 3 or 4 CDMOs, and it's easy for you to transfer your process. So that simple is the context that I want to put it in here as regional manufacture. So make your process and the flexibility and the facility so adaptable that you can switch on with multiple products, also to help you manufacture your products at multiple sites across the globe. Speed, that's ubiquitous. Every unit operation, you need this. And what is that that will help you with speed? So we know process development takes a lot of time, and high throughput devices are some things, and data management. So or predictive modeling tools. So these are some things that you can utilize at the early stages of your process. Involve a lot of sensors and analytical technologies, build in them in the early processes so that it's easy for you to come up with a very robust process and faster. So that's key. Third, quality. So the topmost thing that we are looking at. So when I say quality, it also is not something unique. It is there at every stage. So there is this approach called quality by design, which is more of that actually tags all these unit operations into one place. So take that kind of an approach. Look at approaches or look at tools that use to design your process, and then analyze your tools better so that you understand what is the design space that you're working for each of your unit operation. Deep dive further to look at your sweet spot inside your design space. Look at which is your sweet spot. So that's where you can bring into quality at every step of your process. And finally, the cost reduction. So we'll talk about cost reduction in the upcoming slides when we deep dive a little further on the process itself. Okay. So where are we heading? Are we there already? Are we still lagging somewhere? So we are there actually. So it's quite paced up, I would say. So if I have to put it into 3 different aspects, in the perspective of titer, volume, and cost of goods. So earlier we know processes were at milligrams. Now we know it is in grams. So I've just put greater than 5 grams per liter is what processes that we look now. We heard about processes with 8 grams, 12 grams, 15 grams per liter. So there are indeed processes. So on an average is what is greater than 5 grams per liter. So yes, we are heading towards more improved processes than just increasing the volume. And that takes me to the next point, volumes. So why do we had to go with high volume bioreactors earlier? Because we didn't have yields, right? 0.5 grams per 0.5 mg per liter, 1 mg per liter, very, very, very low yields. So the only choice by then was to increase the volume, right? Go to higher volume so that you will be able to generate as much material as you could. But that's not the case right now. So now you see, this is all attributed to cell lines. So, intelligent engineering with the cell lines have resulted, cell lines producing high quality and high yields. So that's where we are in. It's not just the cell line and media, and also the process adaptation that we see, like the typical perfusion process. So when it comes to volume, you still see 2 different types of evolution in the market. On one side, you see single-use bioreactors with 5 and 6 scale coming up. On the other side, you see people adapting continuous processing or adapting perfusion processing or intensified processes, thereby shrinking their volumes, the production volumes. So two different things happening at the same time, which gives you the flexibility to choose what is the large scale bioreactor volume that you want to, where do you want to play, how efficiently you want to use your cost and money. So that's the challenge in deciding the process that you want to play around. And the third thing, cost of goods. Earlier it was high, and the reason is we know where we came from. Cell culture was synonymous to serum at earlier stages. So serum, unpredictability, lot of loss, all these things were kind of accumulating to the overall costs of a process. But now we know we have very well characterized and developed media available. The processes are getting shrink. A very performing media is available, and well understood process knowledge is prevalent across. So this is kind of looking at bringing down the cost of goods at a greater extent. Okay, what we'll do today is so we know the idea based on the need and the drivers. We know perfusion is the go-to for upstream processes in the future. So what we'll do today is there are multiple stages in the perfusion process, right from media all the way to your final formulation. So for today's process, we'll look at only the intensified production bioreactor scenario. We have there has been a lot of buzz in the market on the intensified seed train. I'm sure many of you have already been implementing the seed train concept. So we'll have a step further to see how an intensified production bioreactor process could be. Yeah. So let's discuss on the process, types of perfusion, and then we'll move on to deep dive further. First, types of processes. We all know what a batch process is, right? But irrespective of what is the manufacturing process, you start with a batch, don't you? Right? So batch is the one that will help you understand the kinetics of your process, how efficient is, if you want to do a simple substrate utilization kinetics, cell density versus glucose, for example. So that's where you start your batch. And then slowly you develop on from there, making it a fed batch or a perfusion process. But we do not use it in the manufacture. The reasons are very clear, like low cell density, low yield, and why is that happening? Because you have a lot of nutrient depletion and toxins built up in the culture. So these are the major things that, a batch is something still at the early stage process, and not something that we can adapt at the later stages or for the manufacturing. Second is your fed batch process, predominantly still leading in the market, most of them using fed batch processes. We see fed batch processes well characterized and designed with very, very, very good performing cell lines and media that supports high cell densities. We see fed batch even going about 30 million. So that's a good improvement in the way of using fed batch process itself. But still we are limited with the cell density and the yield. So that's where we shift our focus on perfusion or a continuous processing scenario. So what is perfusion? What do we do there? We constantly take spent media outside. I'll try to use my pointer. Sorry for not using that until now. So what do we do? We take also shear, pump shear, you use it, and then you take your permeate outside and then push back your cells back into the bioreactor, and then add equivalent amount of fresh media inside the bioreactor. By doing this, what we are doing is we are kind of helping the cells with fresh media every time. Your cells will be happy to have nutrients always available. And the second thing, what we're doing is we are continuously kind of harvesting our product, right? Most of the cell culture products are secreted, and so it's there in the spent media, and that's what we are doing here. So that's a typical perfusion approach. Now we'll see what are the differences in the approaches that people take. Starting from steady state, concentrated fed batch, dynamic perfusion, and then hybrid process. I'll not talk about the seed intensification, which is more of to do with high cell density cryopreservation, or perfusion at the seed stage, like you have N-1, N-2. In N-2, you do a perfusion so that you directly inoculate from N-2 to your production bioreactor. Thereby, you kind of eliminate one seed train, one capital investment completely. So this is something that's the buzz, and as I said, many topics on this have been discussed. So what we'll focus today is on steady state, concentrated fed batch, dynamic, and hybrid. Okay, let's see one after the other. So what is steady state perfusion? So what do we do here in steady state perfusion? As the graph says, you start doing a perfusion process maybe at day 3 or 4 when you see that's the critical nutrient limitation, maybe for your carbon source, and then you perfuse until a certain limit. Say in this case, it's like 100 million. And then what you do after that until certain duration. So that's something you need to optimize and determine. Why do people do that? The reason is there could be some cell lines where even with increased cell density, your yield is not increasing. So there is no point in actually increasing cell density further, which could actually end up your process somewhere here and then you would start declining. So rather have the cell density constant and plateau it for certain days so that you get a very good yield at the end of the process. So that's one of the key deciding factors why people do, or how they decide on this plateau phase of cell density, right? So in this case, what happens is your space-time yield kind of utilization of your hardware and also media consumption goes high. Like, imagine a 20-day process where you start perfusion at day 3 with 0.5, 3 days 0.5, and then 1, and then 1.5. So look at the total volume of media consumption. So it's definitely high. And what it results, 20 to 30% is the average increase in the yield, the cumulative yield that I'm talking about, per cell per day. When you do this kind of a steady state perfusion. And how do you maintain the constant cell density here? We have mentioned something called as a cell bleed. Okay, so that's basically to bleed your cells out to maintain constant cell density. So that's more of your steady state perfusion. And what kind of filter we use here is a microfilter, right? Second is the concentrated fed batch. So beware, this is IP by DSM Technologies. So they were the ones who innovated it. And what's it all about? So what we do here is the quick thing that you have identified is instead of microfilter, we use ultrafilter. So when ultrafilter, you have figured out, yes, so you retain your product inside area itself, right? So what is the advantage here? The total media consumption comes down significantly. And second, your product is kind of diluted, and then you should have multiple chains of downstream processing or pull all these things so that your product concentration is much lower. You need a concentration step before you get into your purification, right? So that's going to be tedious. Whereas in case of a concentrated fed batch, you're going to harvest it only one time at the end so that you still can have a single downstream process stream. Only thing is you have to be aware of the sizing of your columns and other stuffs because of the concentration of your product inside. Quick bottlenecks that we see in such strategies are first, stability of your clone or cell line, and second, the half-life of your product. Okay, so if the half-life of a product is not good, holding them inside a tank at 37 degrees for 20 days is not going to work. So those are some crucial things that you need to consider before even thinking of a CFB, concentrated fed batch. Other things to consider also is until what concentration you are going to have your product inside, because at a certain threshold could be possible that your product starts aggregating, and that's an additional step in your downstream processing, right? So beware of all these bottlenecks before you adapt to the concentrated fed batch. But what we see is a 2.5 to even 6x increased yield in such processes. Next one is your dynamic perfusion. As the name says, what is the only difference in comparison to steady state is here you do not plateau it, you just keep perfusing it until it goes on. So you don't control your viable cell density. That's all you do here. Okay, and this also kind of typically yields a similar results as steady state perfusion process. Okay, may not be sometimes where you see probably your yield increasing at this stage, your cells are still healthy for a higher cell density. So all those things are still in there. But in general, what we see is roughly 20 to 30 average. What we get in a steady state is what we get here as well on the tighter front. And then the approach that you should take while developing such processes based on cell-specific perfusion rate. So we have seen that working well with such processes. And last one is a hybrid. So this is the crown, I would say. So this is combining perfusion and fed batch. So what you do is you start perfusion, hold at a certain cell density, and from that point you start feeding. So until this place, it's perfusion process, and then it is a fed batch process. So one harvest and then one harvest. So two harvests is generally what you see. Here your product is completely removed, and here your product is completely retained. So this is one good approach where we see, very good improvement in the yields. Many people adapting. What is the prerequisite for such a process is very, very, very well characterized process. You need to do spent media analysis until the level of even trace elements, which help you in protein production or antibody production, and understand the consumption patterns very well. Only then you will be able to design such a process. Okay, now looking at all those four, what should I choose? What option should I choose? So if I have to decide on what kind of a process I should choose, I actually should ask certain questions to myself. First is, what is the market demand? Second, what is the yield I'm expecting from my cell line, or maybe at the end of my downstream processing? What is the turnaround time, batch to batch turnaround time? Am I planning for a multi-product facility? And fifth, what is the equipment availability? I'll give you a simple example here. Okay, so imagine that you need 100 kilograms of protein a year. You start with a simple fed batch process with the 2 gram per liter yield, and this will, and maybe your batch duration is say around 12 days. So the number of batches that you need to produce 100 kgs is approximately 28. And you will need around 360 days to get this done if you have only 2 KL bioreactors. And the same thing, if your yield is increased twice or thrice, and you use a perfusion process, which is say 18 to 20 days, the total number of days your single bioreactor is occupied comes drastically down from 360 all the way to 180 days. So you see the drastic difference. So you still have 180 days to think of or to use the facility for product 2 or something like that. So those are some key decision factors that you need to consider. And that's where this table will come in handy for you to understand what's going to be the type of process and what are the pros and cons, right? So ask yourself all these questions, and then you decide on what kind of a process you need to adapt. Another quick comparison here. So say in this case, with the CSPR of 20 picoliter per cell per day, and I am targeting say 5 kg in one batch. So you can look at the drastic difference in the volume requirement that comes down. I mean, cumulative yield with respect to the volume, right? So the yield 5 grams 5 kgs you are getting in 900 liter, you are actually getting it in just 240 liter. Or any of these scenarios, you see how efficient these are. If I consider 100% as my fed batch, you see everything is actually on a higher side. So these are some typical calculations along with the economies of the process you do, looking at the capital and operational investments that will help you decide on what kind of a process you need to adapt. Okay, now let's move into the process itself. So what are the key components of a process? So first is the media. We spoke about media a little bit. Second and third are something to do with the perfusion processes itself. So what is going to be my feed rate? What is going to be the rate at which I remove spent media, the permeate? So this is something to do with the perfusion process itself. And this is determined by either doing a cell-specific perfusion rate scenario or converting that into volume PVDs, volume of perfusion media per volume of culture inside. So either way, it is something that you can do. Third one that is very important is your bioreactor itself. So the bioreactor should be equipped to support high cell density. Fourth one is basically the retention device, be it a hollow fiber or an associated with the pump, would be a low shear pump here. So what are the critical things as you see? The pore size, lumen ID, what is the membrane made up of, what is the filtration area, transmembrane pressure, what are the other sensors that are associated with it, be it pressure, flow, etc. So how well this entire setup is integrated is what makes your life easy when you're talking about the perfusion process. Okay, let's take it one step ahead and see what are the key things that we need to look at. Media management. So we do not need a very enriched media in the first place when you do a perfusion process, because you keep replenishing your media. So wisely choose the media there based on the cost of the media also. So choose the media where what essential your cell needs is available. Look at the length of the process, decide DVDs based on this as well. So you don't pay an extra penny just because you are getting marginal increase in your yield. That's not going to help. So look at the robustness of your process, where the cluster of robust processes lie in your multivariate data analysis, and then pick those processes, pick those medias, and develop them from there, right? Second and third are kind of interrelated: fluid management and process flow. So fluid management is basically what is across what is that supports your perfusion process. So you have your bioreactor, your retention device, but other things are there around it, like you have your weighing scales, you have your mixing systems, you have your totes which are holding your media for addition or removal. So all these things will depend on the type of process that you choose. Like I'll give you an example. In a steady state, when you are continuously harvesting, probably you are looking at a different kind of a process flow. You have to take it daily to the DSP line, or you have to put a bigger tote there to cool your process, cool all the solutions like culture, all the permeate for like 3 days or 5 days, and then take it to downstream. So to that extent on the process, day by day is what you need to plan. So have a Gantt chart or something like that to understand where I start, what is the media requirement, and where it is going, and from there you have to take. So decide on the process flow based on the perfusion strategy. So that is going to help. Otherwise, it's going to mess up the entire manufacturing facility. On the hardware side, we know this is going to be a high cell density process, so oxygen demand will be more. So thereby, your flow rates are going to be higher. So build your mass flows capacity higher, and then give the mass flows flexibility. Like you have 6 mass flow controllers in your bioreactor, and each are tagged to a specific gas, and you will not be able to move it across difference. Like you have 2 sparger inlets and maybe 1 overlay inlet. You don't have the flexibility to switch around. That's not going to help, right? And because of the increased flow rates, also the exhaust filter is something that you need to look at, because you might end up choking your filter at some time. So dual exhaust filters are some things by default you should be considering. On the pumps, prefer integrated pumps. Only integrated pumps will help you create a control loop for your perfusion process, and then design the pump flow rates based on the VVDs that you plan for. Like if you have 2 KL bioreactor with 2 VVD process, so 4,000 liter is what we are talking. So calculate duration where you want to replenish this 4,000 liter, and choose a pump accordingly. Even if the vendor isn't able to provide you an integrated pump, get the pump integrated with additional auxiliary inputs. So have those in handy, like 0 to 10 volt or 4 to 20 milliamp. Try to connect a Watson model, a bigger pump in that case, which you can pull inside the control loop. So those are some flexibilities you have to build in your bioreactor. Apart from the hardware, the other one is in case of a single-use bioreactor, it's the bioreactor bag. What's important? Sparger. Why sparger is important? Because that's what is going to, as a combination, sparger and your impeller system and the aeration strategy all put together will determine what is the mass transfer rates that you will get, ultimately deciding what is the oxygen transfer rate. Only then you will be able to make a comparison between the demand and the transfer rate. So that's why it's important to choose wisely on the sparger. Ports, number of additional ports. Being perfusion, you might need additional ports definitely on the lateral bottom side of your bioreactor. So consider those things completely. Third is CO2 stripping. So increase cell density, increase metabolic CO2. So definitely you will have issues of PCO2. So have drilled hole spargers built in so that you can push a lot of air to strip off the CO2 from the culture vessel. Optionally, you can choose sensors. Why I say this optional is When you transfer a process from your process development to your production, make it as simple as possible. Otherwise, it's going to burden your operator. You do all the stuff in PD. Use as many sensors as possible, as many PAT tools as possible. But when you transfer the technology, make it as simple as possible. So that's why I still keep this optional. Even if you have PCO2 or PCD sensors, have them just to monitor. Please do not include them in your control loops. That's going to complicate your process. And as I said, I was talking about integration of other devices. Exhaust condenser, in case it's really high cell density, 150 million plus, and you see a lot of gas coming out. So then probably you can consider a condenser there. And then also bring in a lot of redundancy, right? Be it sensors, pH, DO, or even the hollow fiber. So if you are taking a different approach where you want, where you see, okay, for my process, probably my filter will go off at 80 million, have redundancy built in, right? So moving to the principal question that we have here, alternate tangential flow versus tangential flow filtration. So since a long time, we have been telling TFF will cause a lot of shear. Okay, so I'll give you a quick study where we have investigated on what is that causes shear or stress to the cells when you take TFF. So let's look at a quick overview here. We know what are the advantages of ATF. So filter clogging doesn't happen. That's because it's bidirectional. Low shear because the path length is very short. We know have been adapted to all cell lines in the market so far. And we also know there is only one port that needs to be connected to the bioreactor, which acts both the ways, right? And what happens in TFF? It has its own advantages, like low hold-up volumes, low residence time because the liquid is not passing in 2 times; it's just passing in 1 time. Flow rates can be wider because TFF was something that has been present even before. Flexibility on various filters, and we have known that TFF can be used with high viscosities. Just with a little bit of tweaking, these things can be taken care of. So 2 things that are told as a stark advantage when it comes to ATF are shear and fouling. Okay, for fouling, the alternate is basically to have an additional filter and then use some kind of an intelligent switch so that you put in a condition and then the system should be able to switch it to the second filter. Maybe your transmembrane pressure reaches X value, switch to the second filter. So those are some adaptations that we can talk about. Otherwise, predominantly spoken about is the shear. So let's look where the shear comes from when it is on the TFF system. Okay, so an analysis on CFD specifically on the shear of the cell. Now we know that 100 Pascal, will anything above 100 Pascal is more of not even 100, a little bit more than that. Your CHO cells or other cell lines are a little bit stable for. So quick analysis shows the CFD modeling here is for both bioreactor, the fluid path, and also the hollow fiber. Hollow fiber is not mentioned here, and the hollow fiber is pretty much the same. So where do we get this shear for the cells from? It's mostly the fluid path. So avoid having sharp bends in the fluid path. Avoid having reducers or what happens basically is the red. You don't see much of red area here, but whenever you have reducers like this, you're reducing from 1/4 to 1/8. So that's the place where your cells are getting stressed off. Okay, so once you establish a fluid path without this, we see the cells are the shear factor or the viability loss is very much comparable between ATF and the TFF system. Apart from that, also have these things in handy. What is the shear that is caused by your bioreactor itself? Shear factors in terms of impeller shear, bubble shear, as well as Kolmogorov eddy shear. So have all these things handy so that you will have a good understanding of the process that you develop. If it is a single-use bioreactor that you are talking about, get these from the vendors that you get the bioreactor from. Okay, so quickly, how do you simplify your perfusion process? We know there are multiple things involved. How to simplify it? First, make it more of a plug and play kind of a device. Use rapid setup. So you have a hardware with integrated pumps, pinch valves, and stuff like that. just put in all the fluid path or get a ready-made fluid path with the pressure sensors or flow sensors, etc. integrated. That way you don't have much of a manual interruption in the assembly. Second, eliminate unknown parameters. So how do you do that? Have sensors, as many sensors as possible in the in each line, be it potentate permeates. Have a lot of sensors so you get a good understanding. There are people who also have additional sensors integrated, like online measurement of glucose. That could be a trigger for your perfusion process. So those are some things that can be thought of. Third is minimize manual failure risks. So when does this happen? So most of this failure happens when you kind of interrupt with the system at certain point. Even when you build in redundancy and you need to be there to switch the filter, probably that's one of the places that you can attribute a problem for. So make it as much automated as possible so that you avoid all those errors. Decrease stress on cells, as I said earlier. Reduce the fluid flow path, avoid sharp bends, and also reducers. So finally, as you see, yield, economy, quality, robustness, these are all in multiple directions, multiple planes in a given space. So what does perfusion does is actually brings all these things together at one point. But that's not so easy as we were talking on since the beginning. So a good characterized process is important. So that will help you understand the limits of your process. A QBD approach will help you design a key attribute in the entire unit operation. And then with improved cell lines, improved processes like the perfusion, you will definitely end up improving your yield and thereby robustness of your process. So robustness is basically to simplify the equipments associated with all these stuff, and then you transfer a very, very simple process to the manufacturing. So you have entire control, you know what is there in your process, what you have developed, and that way you will have control on your process also. So apart from upstream, what are those stuffs that are actually, it's more of a cascade effect, I would say. So based on the process, as I said earlier, based on the type of upstream you determine, your downstream will vary accordingly. One harvest, different downstream. Two harvest, three harvest. So that's a different downstream. And that brings you to the third point, which is batch definition. That's an important factor for a perfusion process on how are you going to define your batch, because you still have one batch for upstream and multiple of your downstream. So what is that you are going to define as batch 1, 2, 3? So these are some things for you to think about if you are looking at envisaging into the perfusion space. So and finally, the risk assessment part. Typically, like any other processes, look at all the other additional stuffs that you have brought into the unit operation as the key factors for risk assessment. Yeah. So to conclude, what I would say is look at all, have all these tools available in the PD space: sensors, PAT tools, data analytic tools, and build in a very good cell line and media. Then you are, and while developing your process, do spent media analysis pattern, then you'll end up developing a very, very good perfusion process, and that's going to help you in the long run. Yeah.
Thank you very much, Vasudevan. That's a lot of material to get through, and we have a lot more to come. So there are no questions for you currently in the chat, and we're running a little bit behind. So if I may, that's okay. If I may, let's hand over to Ian so that he can continue. Ian, are you okay? Great. Yeah. Thank you, Ian. Over to you. You need to unmute.
Yes. Thanks, Linda. Hello, everyone.
On your slides again, we seem to have lost them.
Oh, my goodness.
That's okay.
Is this, you're seeing something suitable, are you?
We're seeing your desktop, I believe.
Oh, good heavens. I beg your pardon. Oops.
Yeah, that's perfect.
Is it right? Okay.
Thank you.
Multiple monitor confusion. Okay, hello, everyone. Let's get started on part 2. I've been asked to talk to you today about continuous chromatography as a possible option for intensifying future downstream purification processes. It's just, as you'll be aware, one of a number of options, and I'll be going through it rather quickly. It's a fairly complex topic, so I'll leave my email in the chat at the end. If you want to get in touch with me about any clarifications, any questions, please do. So, without further ado, here we have a crude depiction of a protein expression process in cell culture, showing a number of large tanks for large bioreactors, suggesting a staggered output to feed a downstream train. 4 columns, that could be one large column for capturing the protein from the bioreactors, or it could be one column, smaller column being multi-cycled, the product stored in an intermediate product tank between every step, big tanks, process buffers feeding into the system, again from large prep tanks and hold tanks. Again, this is all contained in a large purpose-built clean room, every square meter of which is costing you money. So to meet the future need to reduce the cost of therapeutic drugs, there is a lot of pressure to reduce the size of clean rooms or get more productivity from every square meter of existing ones. So one tool for this is continuous chromatography. So it's just a picture of continuous chromatography systems here. They look very like the standard benchtop chromatography system, quite like a standard skin-mounted chrome system for manufacturing scale. And here we have a key statistic. We have customers who found, or there are customers in the industry who found they can decrease their total resin volume for a chromatography step by 30 to 50%, or reduce 30% process time, which has knock-on effects in capex savings and footprint for everything up to and including your buffer prep, your hold tanks. It has some advantages. There are also challenges. Running continuous processes are intended to run for long periods of time, days perhaps, eventually weeks, and eventually months, possibly years, which raises new challenges in terms of bioburden control, and you certainly want to be able to CIP the thing during the process. An ability to handle variations in feed and column volume. If you're packing your own columns for a batch process, you can tolerate a variability in the bed volume. For a continuous chromatography process, which uses multiple columns, you need to be able to handle that variation in the number of columns you may use. The scale up from the lab may not be exactly as we know it now with the batch process. And variations in feed. If you're running for months, you will undoubtedly find the tighter varies in your feed. And a good PCC system should be able to deal with that. So just to be sure what we know what we're talking about with PCC, on the left we have batch chromatography being depicted, where all your chromatography steps are serial. You load the column, the one column usually, 2 or 3 if you're running SEC chromatography. Then you wash, elute, strip, regenerate. It's all sequential. In a PCC system, you have multiple columns and multiple active process flow streams. So here's a simple depiction of continuous chromatography. One feature is that you will be loading 2 columns at once. I've shown 2 here. Some PCC systems actually have multiple columns, more than 2, but for simplicity here we show 2. So if we look at this step 1 on the right there, you can see that column 2 is being fed with sample, and it's being fed with sample until it starts breaking through at the outlet and flowing onto column 3. While that's happening, column 1, which has previously been loaded, is being washed and eluted. Column 4, which has previously been washed and eluted, is being regenerated. And it goes through, with a 4-column system, it goes through 4 steps. And column 2 is loaded to the level you want to load it to. It's washed and eluted. Then you start directing the sample to column 3. the clean column from the previous step is put in series with that column to catch the breakthrough. Column 3 becomes loaded to the required level. That's eluted. The clean column from step 2, which is column 1, is then put in series with column 4, and so on. It just works for a 4-column system. It works through these 4 steps. So for 4 columns, you have 3 active flow paths. I'll show it a bit more readily on the next step. So this shows 3-column and 4-column. So in a 3-column system, you've got one flow path is loading 2 columns, and one other flow path using 1 or 2 pumps is eluting and regenerating. A 4-column system, one pump is loading 2 columns. Another pump is regenerating a column that's being eluted, and another pump is washing and eluting a 4th column. So it just basically goes through this cycle repeatedly. So the challenges for PCC are that with a batch column, you can control the column loading. You do your experimentation and process development. You flow sample through the column until you find unbound protein in the column outlet. You maybe continue till you get 20% or 10% or 20% unbound protein in the column outlet. You fractionate everything you recover from the column, and you work out how much protein that column can bind at that flow rate. That's all well and good. So then you can calculate how much volume to apply to the column to bind the required amount of protein. So with a PCC system, you have an issue that you have multiple columns, perhaps 3 or 4. If you pack them yourself, there's going to be invariably some variation in the volume and their ability to bind protein. The total binding capacity in grams will vary. If you're running for extended periods, taking the contents of more than one bioreactor or one bioreactor operated in perfusion mode, your feed titer is going to change. So you want to be able to load that column consistently. So you can do your process development and just work out a volume to apply, but there are better ways of doing it. So one way of doing it, which is implemented by a couple of companies in the market, is to use UV detectors at various points in the flow path. So if we look at this diagram top left, this should help to understand dynamic control, as it's often called. So what you would normally do is have multiple UV detectors. You'd have one in the feed stream before the first column. You'd have one after the first column you're loading before the second column. And you'd have a third detector after the second column that's being loaded. You'd also separately have another UV detector monitoring the elution of a column in a different flow path. And you may or may not have a UV detector monitoring the regeneration. But if we look at this top left diagram, we can see how the UV detectors in the load flow path, what they detect. So if we look at this brown line here, this is the sample UV detector. So you can see what happens here. X equals 0, that's the pump starts. Shortly afterwards, absorbing materials detected in the pre-column detector quickly goes up and plateaus at a level which corresponds to the UV absorbance of the raw sample. That's your target protein and the UV absorbing impurities. So the red dotted line would be the UV detector after the first column to be loaded. Now you can see here there's a delay before that rises because the sample has to work its way through the resin bed, and then when it does so, the UV in the column effluent rises and plateaus at this level. So at this point, where this plateau occurs, this is the UV absorbance of the UV absorbing impurities. So that's all you see in a batch column. You wouldn't want it to go any higher than that. You don't want protein breakthrough. But in a PCC system, you can see as we carry on, the UV rises again. That represents protein, unbound protein appearing at the column outlets of column 1. And if you allow that to continue, it rises and then plateaus again at a point equal to the absorbance of the raw sample. So you've got complete flow through, no bindings happening. So the difference between these two plots is the impurity baseline, as it's called, and the absorbance of the raw sample. That represents the absorbance due to your protein. And if you can measure that, you can control the loading of the column to a point which represents a percentage of that delta. So the first interesting point, PCC compared to a batch column, is you have to be able to measure the absorbance of your raw sample. Normally for a batch process, you don't typically mind if the sensor saturates. PCC, you need to be able to measure it. The second thing I'm going to make, I have to move quickly on this. If we look at the bottom left, you can quickly see that you imagine a process running for weeks or months. You're not going to be able to home in on every elution peak, overlay them, check for comparability the way that a supervisor or biotech will in the current day. What you're looking at with all these dozens or hundreds essentially of small elutions is you're looking for trends. So you're relying heavily on the automation to spot trends and things like column DP, elution volumes, anything that might signal a problem. So this is pairs very well with automation. Automation's key in making it work. So this is just, I'll quickly go through this. So these figures are taken from a paper published by Merck and GE in the Journal of Chromatography about 6 years ago, in which they managed to demonstrate that the purity, the actual function of the PCC chromatography compared to batch was very comparable, up to 31 grams per litre. That was 6 years ago. They found at the limit they could achieve 60% decrease in total resin volume. That's the sum total of the resin beds spread amongst your 3 or 4 columns, compared to one batch column, and they achieved a 280% increase in affinity productivity. Again, that would be an extreme, and I would have to say that you can't have both of these targets at the same time. You either optimize your PCC chromatography for resin capacity utilization or for productivity, or for an optimum between the two. But if you chase one of these goals, it will be at the expense of the other. So this is just, I'm going to skip over this one. This is a bit of a Cytiva marketing slide, I'm afraid. It says nothing we haven't said already. So here we have a picture showing the resin beds. You see from these axicron columns here, the resin bed looks quite short. Now, if you think about it, you're loading 2 columns in series. If you normally use a 20 centimeter bed and you try to load 2 20 centimeter beds in series, you'll for sure run into pressure problems or have to slow down your process by slowing the flow rate. So typically, PCC columns use shorter beds. This looks something like 10 centimeters. But this has interesting implications for scale up. Because normally when you scale up a column, if you have a 10 centimeter bed in a laboratory column, it's maybe 2 to 5 centimeters in diameter. It's a column. It's a long tube full of resin. If you scale that up to 2 meters, what you have is a very flat-looking disc. And to get a good homogeneous bed with no weaknesses that may develop channels, it requires good resin distribution in that column. But with PCC, you're not scaling up to 2 meter columns. A PCC system using 3 or 4 columns, the biggest columns I think we see modeled at the moment are 60 to 80 centimeters. So you can potentially actually use shorter column beds than you would with a batch process, at least one that was intended for scale up. So that's an interesting thing to note. So these systems have been built. I'll just quickly summarize this slide. And we have seen them able to handle the variations in feed concentration and disruptions from upstream. They can be cleaned in process. We have resins which are caustic tolerant now to 1 molar. The larger systems with half-inch piping can actually be steamed from time to time. And they can be integrated well with the DCS. And the larger systems we've seen running for 24 hours or up to 20 to 30 days is the limit that we're aware of so far. And it can be operated with a DCS and linked to upstream and downstream unit operations, or used as an island of automation. Okay, so just a quick word on scalability. So this is what we look like. We have systems on the market that will work on the bench with a few litres of liquid, going up to a half-inch system, which is that's the pipe diameter. All the pipe diameter you need to marry to a 10,000 litre bioreactor. So this picture on the right, for you as engineers, I'll just explain this is a very early design. You'll appreciate with multiple columns in use, you need to keep the flow path, whichever flow path is selected, you need to keep the actual flow path length the same, and you particularly want the distance from the column outlet to the downstream detectors to be the same as well. So the original systems, that was realized by making the chromatography systems a polygonal shape. So you can see this 3-column system has a hexagonal outline. It's very large. These days, our engineers have managed to achieve the same aim within a much more compact, traditional rectangular footprint. So the reason this large, older system is shown is that there's a lot of innovation in the PCC field. Our systems are all custom systems, and customers often ask for very interesting and inventive features to be incorporated, which means we can't show you a current 1.5-inch system. So this is just a paper that was published by one of our partners, Merck of New Jersey, just reporting results they had with integrating 3 unit ops with the PCC system. Integrated protein A capture with anion exchange chromatography and virus filtration. They achieved a 2 to 3-fold productivity increase with the PCC alone compared to batch, and a 2-fold productivity increase for the 3 linked unit ops. And again, they talk about the use of process analytical technology, because as mentioned earlier, that really is key to making connected processes and eventually continuous manufacturing work. Okay, so just quickly mention half-inch systems. We're used to seeing those used for early stage clinical manufacture perhaps, but here a half-inch PCC system with a titer of 5 grams per litre could potentially purify 20 kilograms in a 24-hour period, which is quite an impressive figure. Okay, we have, I'm going to have to go very quickly here because I'm robbing Jai of his time. So I'll just quickly run through some potential uses. So here's another example of scalability PCC systems. A 50-fold increase has been demonstrated within the same PCC system for map capture, from going from the lab machine to a 3/8 inch manufacturing system. 294-fold scale-up has been demonstrated. And I'll just quickly mention application examples. We've talked about MABs, but it's been used for recombinant proteins. Vasu talked about unstable molecules. PCC adapts very well to that. You can start purifying your material as soon as it's harvested. That gives you more control over unstable molecules. It has been used for vaccine processes. Sanofi have done work with our PCC systems. So they used SEC on an E. coli process. It's also been used in plasma fractionation, purification of plasma fractions. And I think basically I'm going to go very quickly through this slide because we really are getting short of time. So this is an interesting diagram, this pentagon. So you see here we have batch process in orange, 3-column PCC in solid blue, and 3-column PCC non-optimized in the dotted blue. So you can see the batch process uses a lot of resin, uses a lot of buffer, moderately good on capacity utilization, okay on process time, okay on productivity. So a non-optimized PCC system, if you just took a batch method and turned it into a PCC method, you could very easily get the situation of this blue line here. Your total process time could go up. You'll save on resin volume probably. Capacity utilization, you'll see some increase. Buffer consumption will go down. But productivity could even be less than the batch, which is interesting. But if you optimize the process and you get all two or three or ever how many of your process streams taking the same amount of time and those columns switching efficiently, an optimized PCC process or process that was designed for PCC can increase your productivity above batch, reduce your buffer consumption, increase your resin capacity utilization, reduce your resin volume, and reduce your process time, or at least increase it slightly. But it's not a given. If you translate a process to continuous chromatography, you will not necessarily realize all the benefits you expect unless you optimize the process for PCC. It's a big topic, so if anyone wants to go into that with me, I'm more than happy to get in touch and go into the details of it. Okay, so I'll just wrap up with the pros and cons. The pros: increases production rate, equivalent resin volumes, reduces the required resin volume at longer process times. So that's what I said about the trade-off between productivity and resin capacity utilization. You will get a bit saving in buffer consumption because your total resin volume is less than the batch column. And processing of unstable molecules is beneficial and well suited with continuous loading is required. Less suitable when short process times are required. For clinical use, it probably doesn't make sense if you just want to make 3 kilograms, say. The setting up of the PCC system and the writing of the method, developing of the method, probably doesn't make sense until you're getting into long extended operation. And less beneficial at low titers. If you have a legacy process with maybe 0.5 gram per liter, the load time will just dominate the purification so much that you probably won't see any benefit at all. And not suited for scenarios where the process doesn't reach steady state. Again, that's where you get into the continuous 3 or 4 column loop, do one loop and then shut down again. It's not worth it. Okay, so acknowledgements to our partners, and thank you for that.
Thank you, Ian. You're not off the hook yet. We'd just like to ask one question, which was posted by Bankin Oza. When you use actor PCC system generally, what is the delay volume when you use UV detector? Is it the same without the UV detector? And I'd also ask Jai to load up his slides while you're answering that.
The delay volume to the UV detector. The larger systems are designed, as I said, that whichever flow path you're using, the distance from the column outlet to the detectors is the same. It's a necessary design parameter of the system. Is that the question that was asked?
Bankim, do you want to unmute? Are you happy with the reply?
Yep. Hi, Ian. This is Bankim here. I mean, what I wanted to understand, is there any kind of significance of the delay volume in the entire process when you are using the ACTA PCC system, or does delay volume play a significant role, or is it just on the parameters that are there? I understood your point that UV detector is the prime detector that is used in these such kind of scale-up processes, but does delay volume, just to make it simple, does delay volume have any kind of significance as such?
It does the same as in a batch process. And if you look at the delay volume between the downstream detector and the catch vessel for a single column chromatography system, the same information will apply to a PCC system. It will have one delay volume between the detector and the outlet, whichever flow path is chosen. So you take it into consideration in exactly the same way. There will be one figure for the delay between the downstream detector and the outlet.
Okay, thank you.
Thank you both. If you have any other questions, please do post them in the chat. Even if we run out of time later on, the questions will be forwarded to the speakers so that they can address them in more detail with you personally. So without further ado, let me ask Jai to share and thank you.
Yep. Thanks, Linda and Ian. Yeah, so today we'll be running through on the advanced process technologies and pharma digital IoT. So the main scope of the talk here is more on the digital side and automation side and not on the process side itself. So there's a big question usually multiple people usually ask is what is the difference between automation and digital, right? So is it just a fancy word, digital? Is it just a marketing stunt? No, if you just really go in detail and understand what exactly is the difference between these two, that is where like let it be just machine, individual machine without automation and with sensors, or if it is a supervisory control and data acquisition with PLC or DCS as such. So and MES, manufacturing execution system. So all this would still be under the automation purview, but when it comes to digital, you can see there is a clear differentiator here being the cybersecurity aspect of it. When let it be in a GMP process, for example, does cybersecurity is not of a concern in SCADA or PLC? Definitely it is, but it is more of a localized network. So it's not going to get broadcasted to the outside world or the internet as such, or through the DMZs or anything as such. So whenever there is connectivity outside of the plant area to something like a cloud solution or IoT solution or anything as such on data historization through cloud, so that is where it all comes under digital aspect. And any plant for that matter in the manufacturing process, like which are the levels it's going to be staying at, right? There are 5 broad levels, which is like pre-digital plant. We will be just using some PLC controls, standalone controls, something like that. And level 2 would be on digital silos. So each system controlled with just one automation software, not the other system. So connected plant would be something like PLC or programmable logical controller, where multiple instruments are controlled with one particular automation system. The predictive plant is where the automation itself predicts on what is going to happen. Let it be like preventive maintenance or these kind of things. And adaptive maintenance is not just predictive, it is adaptive. So how it's going to happen now. So if there is a alarm which is generating because of one particular reason, how the process is going to get adapted to suit the continuous flow of our batch process. So these are top 5 levels which are actually categorized into as per the Bioforum. So on evolving biological industry and drivers of change, I'll not go through in detail. These are very much explicit, and we all know from the industry that there is a need for change, not just on the process side, not just on the hardware side, but the automation and digital concepts too should be in the same pace as in the other areas or domain where the molecules are being discovered and vaccinations are being developed. So are we meeting all this now? Definitely yes. The industry has come a long way in this aspect, and there are so many improvisations which is happening. And what did COVID-19 make into the digital era? It has really speeded up more processes here. To be frank with you, I represent service team here. So there are so many installations, let it be servicing, which is happening through digital technology, through cloud solution, remote assistant tools, remote alarming tools. So these things are predominantly evident now. That is what customers are wanting now. So this is all just thanks to the COVID-19 in this aspect, where the digital era has come to a much faster pace than it would have happened without it. So coming to a few examples on advanced process technology, like I'll be running down through 4 solutions here, or the problem statements itself. So coming to one of the solution, it is like pre-designed and productized configurable automation platform. When we speak about automation, there are n number of companies who provide automation solution, and not just integrators, I mean, but the OEM itself. We have Wonderware Software, we have Rockwell, we have Omron, we have whatnot. Like there are so many companies who are offering, but the when there is a scientist who is still developing in part of the PD, and they have the roadmap to go into manufacturing. So how it's going to help on the automation in reducing the time and cost if they choose the automation in the right direction in the stage one itself. So this is where the solution is going to come handy. So the code base itself is going to be pre-designed and productized, right? So once we pre-design how it's going to be configurable, right? So both are contrasting terminologies here, but really this is the way industry is tending towards. So a standard kind of solution which caters for the standard offering and connectivity with the system, and not just that, and even configuring with any other new instruments or new tags. So this is where the industry is tending into. So something like the figure eight platform what we offer, so it the same code base which is being used for a 50-liter bioreactor, the same can be implemented for 200-liter bioreactor, or the same can be even implemented for a 2000-liter bioreactor. Similar concept works for all the units as such, right? So that is where the industry is tending towards. So what are the benefits of it? So like with this, like it is easy to manage, no need to redo the code base right from scratch, no need to invent the wheel again, and no need to even qualify to a greater extent here. There are fewer qualification steps which would be required, because the design is already qualified, only the system needs to be qualified. That is where it's going to add value. And it has much more this configurable automation platform with standardized library. This is going to be adding value for scale up as such in reducing the time. Yeah. So from our study or from our practical experience, right? So with this kind of solution, like 7 years, if it's taking to get into the CMC timeline, right? So it's going to get shrinked to 3 years. So within 3 years, all the hardware or the automation side of it will already be ready. So this is where it's going to be adding a significant contribution or KPI to the organization as such. Yeah. So as I just mentioned, like it can be just a simple benchtop bioreactor to the manufacturing scale 2000-liter bioreactor, to give an example. Same thing goes with all the bioprocess steps in the manufacturing. Yeah. So the second problem statement would be something like on analytics-based process simulation tool. So what would customer usually want to is they if they are running a smaller scale bioreactor, and how are the parameters or the variables attended or related in this aspect is going to be behaving if the volume is going to be increased because of their scale up. So that is where uh, the scientists have to really work on how much is going to be their KLS, how much has to be their gas flow, how much has to be the agitation speed. So the solution here is actually on, we have a tool which is based on analytics as such. So enough data is filled in there using ANSYS simulation models and even the practical trials being done in the physical systems. So based on the total number of days of culture and what are the volume they're expecting and how it is done now. So based on these inputs, they would be just getting the exact figures and variables of having the typical and most suited parameters for their process. The third problem statement would be like a cloud-based PD data management tool. So usually customer would ask, like I'm spending more time in my on the data itself, right? So shifting the data from manual, again sharing it, again reviewing it, and again validating it. There are so many things which are involved here. So this kind of a solution where it is a cloud-based data management application, more like a historian maybe. So how it's going to address? So this is where the cloud-based solution is being used, not just by us, but multiple industry across the industries as well. So here what happens is in the PD, whatever the data is logged or generated, this is all going to be looped into a larger historian provided through cloud solution. So again, there is a more important point here to combine the data and reduce the time in managing the things, and the data itself is secure as well. The next problem statement would be like, this is more of CGT, cell and gene therapy portfolio. There are so many manual processes which are involved in the current day GMP processing for CGT. So there is no proper solution where the data is handled with electronic batch record, with whatnot. So because GMP and gene therapy cell building is not that straightforward. So as the number of patients being treated using the cell therapies is increasing day by day, so this is going to be adding a real value when we have a cloud solution with unified digital platform where all the instrumentation and CGT are connected to one single solution. So that is where this would happen, where we have one kind of a cloud solution. So it would contain the batch record. It can release by exception ESOPs, so manage the operators or user management as such, and equipment connectivity and integrated logistics as well. So in this kind of cloud solution, what the industry can expect is where are the cells being transferred at. So there is GPS tracker which are available when the material is being shipped or transferred from one region to another, because this is directly related with patient, right? So the other part, second half of my like topic is more on pharma digital IoT. So uh, in general, when it comes to serviceability and maintenance, so how the industry asks us, right? So I need to know which instrument is operation and available, and I cannot lose my experiment or sample. Can there be a proactive servicing, and can I predict the unplanned downtime? Or can I get the immediate response or alert to the service engineers from the OEM supplier or anybody as such whom I assign them to be? So wherein they can come in, pitch in, and give me the immediate response, let it be physical or through some digital tools through augmented reality. Or something is wrong in my process, how can I quickly inform the supplier as such? So this is where the IoT comes into picture. So the customer would be expecting to have the data real time, proactive, so that they have their utmost uptime to accelerate their processes as such. So, but the must is here, they, the data should be secure, protected, and connected, by which they can achieve their efficiency even in the GMP process. So the traditional approach, how the serviceability of the products used to happen in biopharma, is just on call or on through email. So the turnaround time is going to take more. If it is a specialized machine, it's going to take much more time as such. So that is where the data-driven technologies are being implemented across the industry. So the life cycle management of the product itself or the instrument itself is catered to provide the maximum uptime to the customer processes as such, and have a long-term relationship with customer. So the tools and process as a culmination uh, like leads to the expertise of the company and the technology itself. So one very good example I would like to give is like, take for example our instruments. So these are all connected to one network, so which would be going right to the IoT gateway. So this would be again a PowerEdge server, which acts as a IoT gateway, which will be the bridge between the process and the cloud solution as such. So once it has connected to the cloud, so there'll be an online center or a technology. Using the technology, there will be a center who will be monitoring these things, and not just manually, but automatically it would be assigning to the corresponding field engineer team or the services team who will be addressing this issue. So the first option would be to remotely monitor. So within the times are like within 1 hour, the remote engineer should log into the issue and log into the issue by sharing the customer session and glancing through what is the issue. And even before customer knows there is an issue, through the IoT solution, the customer would be alerted. There is some problem with your UV lamps. This is the time to keep a spare handy now. So something like this, a culmination of technology, process, and people. So this is where the industry is heading into. And coming into the cybersecurity path, so all the IoT solution as such is very much aligned with cybersecurity, and the protocols are IEC standards as such. So the main challenge what the solutions would usually speculate is on merging the OT and IT network. So many customers, even I have discussed with, when we merge OT and IT, even with the proper DMZs, dematerialized zone or firewall in place, it's not going to be so straightforward and easy. So this is where the cybersecurity aspect has to be considered in any IoT solution in the GMP process, by which customer will get the efficiency in their process and manufacturing. Yep. I think I went pretty much in fast mode, provided the time consideration. Please let me know if any questions you have at your end.
Thank you, Jai. Thank you for racing through that. There's a lot to take in. Does anyone have any questions? Please post it in the chat and we will ask the speakers. Feel free to unmute yourselves as well if you prefer to just state your question.
Hi, Jay. This is Bankim once again over here.
Yep. Hi, Bankim.
I just wanted to understand when you showed that slide that multiple systems of different brands can be hooked up on this cloud. Will that still be under the compliance of 21 CFR, or how does it go?
Yep. So if you see here in the gateway, there is a clear dematerialization between the OT network, the operational technology network, and the IT network itself. So this is under compliance. Like, for example, AWS, right? They have come up with the life sciences portfolio in offering the solution. So the industry is really tending towards these particular things. And as I said, usually customers will have a real worry when IT and OT some or the other way is getting merged, but it is under compliance. Yep.
Okay. And second question, just to add to it, like the cloud computing we are talking about, is there additional backup that is done on some kind of supercomputers, or like just data savings, or any kind of crash that happens?
Yep. This cloud solution will be a live cloud solution. So the tags which are read from the instrument will be in the proprietary native technologies like OPC or Modbus or whatnot, like the industrial protocols in general. So once these are pushed to the IoT gateway to the cloud, so since these are live instantaneous values, so the enough redundancy or backup, recovery, and restore options will be implemented in the cloud solution itself. Coming to the systems itself, so the database or the tag monitoring or even the control of the system will lie in the OT network itself. So the control and data logging part is independent with the cloud solution itself. So the cloud solution would only be complementing on the data which is being generated from the processes.
Okay,
that's a good question. Yep.
Yeah, just to summarize, just to understand, the user, if the user is remotely working on a company-based network, it's only then he or she would be able to remotely monitor this entire process or get the alerts, or is it something that the user has to be basically on the network?
Yep, that's a very good question again. When it comes to the processes here, so it has to be not just the company domain, but the OT domain. So the user has to be in the OT network. So he'll not be able to operate remotely. In general, tech solutions is that way, though we can make it remote as well. But when it comes to the connectivity here, they can connect from company provided assets. Usually they prefer that way in their domain, the company
Right.
or the customer's domain, through which they can connect to the IoT gateway. But technically, this is again just a AWS server, so we can just connect it through any means as such. But provided the GMP background, I think that is where customers don't want this to happen. Yep.
Okay, thank you.
Yep. Thanks.
Thank you for your question. We have time maybe just for one final question if anyone wants to ask. In the meantime, we've also posted up a link, which we would be grateful if you could spare just a few minutes to fill up and give us some feedback so that we can improve for further sessions. As mentioned in the chat, the link to the recordings and the slides will be made available as long as we have your email address, which you use to register. So we don't have any other questions right now. I think that you've given everyone a lot of food for thought, gentlemen. Thank you all for your time, Vasu, Ian, and Jai. And speaking of which, I guess it's almost dinner time, and we want to wish everyone a good evening and stay safe. Thank you for joining us, and we look forward to seeing you all again.